And I'm Jessica. Welcome to our show.
Jessica and Ronnie.
Take Biology. Today's topic takeover is the revolutionary form of gene therapy, CRISPR.
But first, you guys know the deal. We're going to call one lucky audience member to come on stage and answer some questions to win a few prizes.
Okay. Who's going to be the lucky audience member today? Let's have you with the black jacket come up to the stage. Okay, let's have you introduce yourself.
What's up, guys? I'm Casey.
Nice to meet you, Casey. For today's prizes, you have a chance to win a mountain bike, a smartwatch, and an overall A+ in your biology course.
Let's get started.
Genetic disorders can be caused by a mutation of one or more genes in an individual's genome or by the inheritance of a faulty gene from your parents. These disorders affect many people around the world, and there are between 4,000 and 6,000 diagnosed genetic disorders. Historically, these disorders have been treated through classical gene therapy techniques that are used to supplement genes.
Okay, Casey, are you ready for your first question?
Yes.
Can you give us 2 examples of genetic disorders?
Sickle cell anemia and, hold on, it's on the tip of my tongue.
Time's running out.
I got it. Down syndrome is another good example. It's a single gene disorder like sickle cell anemia would be easier to correct than a complex multi-gene condition like Down syndrome.
You are correct. Great job.
Now, however, gene editing is being explored as a way to change an organism's DNA. One form of gene editing uses clustered, regularly interspaced short palindromic repeats, or CRISPR, which is a DNA sequence that encodes an RNA used as a guide for the Cas9 enzyme to cut DNA at sequences complementary to the RNA sequence. The enzyme Cas9 can be programmed to cut any region of DNA using an engineered guide RNA sequence. Using this CRISPR-Cas9 system, scientists can remove defective sequences and insert beneficial donor DNA.
If scientists want to silence the gene, they allow the cell's own repair mechanisms to try to repair the cut. This process is error-prone and usually leads to a mutation that disables the gene. Scientists can also replace mutant genes with a healthy copy by adding a piece of donor DNA that contains the new, healthy version of the gene. One of the main applications of CRISPR is that it can be used to cut out a specific mutation identified in human diseases. This could be groundbreaking because the effects of genetic disorders could be alleviated by correcting and replacing gene mutations. A 2018 study on the applications of CRISPR technologies found that they are also being used to increase livestock breeding rates and create new pharmaceutical drugs. Also, a 2017 study published in Kidney International described the generation of polycystic kidney disease in rats. By discovering what genes cause certain kidney disorders, scientists can discover how to prevent these disorders in humans. And in agriculture, CRISPR has many possible benefits such as better nutrition, disease resistance, drought tolerance, and higher yields in as little as 1 to 2 cycles of the CRISPR-Cas9 therapy.
Okay, Casey, this is the last question. Name one downfall of using this technique.
A change to the germline can be passed to the next generation.
You're correct. Germline editing could lead to designer babies.
If we're able to genetically modify our human embryos and reproductive cells, what will be next for the human race? We will be able to include our desired traits such as eye color, hair texture, level of intelligence, athletic ability, and so much more.
It seems like there would be no limit to what we could or would do. Who or what would regulate this? Who would be assigned to make the ethical decision on what should be included?
As of right now, that has yet to be determined. As we make advancements in the medical field and technology, I believe that there will be a system put in place to help us make the right decisions to save as many lives as possible.
Another downside could be the possibly dangerous off-target effects of CRISPR. The specific sequence targeted could be found and cut in other places in the genome. This could cause major issues like disabling a tumor suppressor or even activating a cancer-causing one.
Scientists are still trying to figure this one out on how to determine how many off-target sites are altered with each CRISPR therapy. Although there are a few downfalls of this approach, the pros definitely outweigh the cons.
If CRISPR is approved to be used in gene editing in humans, it could have potential life-saving applications and be able to improve the quality of life for those suffering from chronic genetic disorders like sickle cell anemia and cystic fibrosis. Any final questions, Casey?
Yeah, just one. Can CRISPR be used to not only stop chronic diseases but also deadly viruses such as HIV?
Yes, within the year 2015, a scientist they used CRISPR to cut out the HIV virus out of living cells, proving that this mechanism could be used in the future as a cure.
This procedure was then followed up with an experiment on rats that had the HIV virus in about 99% of their blood cells. With a simple insertion of CRISPR, they were able to remove 50% of the HIV virus from these rats.
I've learned so much about CRISPR today, but I have one last question. What does it stand for?
Clustered regularly interspaced short palindromic repeats.
Just remember, CRISPR was adapted from the defense system of bacteria.
These bacteria, mostly archaea, gain immunity from viruses by cutting specific sequences contained in the nucleic acid that viruses can inject into the bacterium.
Well, that's all the time we have for this week's Jessica and Ronnie Take Biology episode. Thank you to everyone who watched to learn more about CRISPR.
And thank you to our incredible and helpful special guest, Casey. Everyone give him a round of applause.
Enjoy your prizes, Casey. Jessica and I will be sure to get you that A+ in Dr. Stevens' biology course.